So here's our question. A 36-year-old man presents with diarrhea and abdominal pain. Just a few hours before, he reports having eaten at a new Chinese restaurant. The only thing he ordered was fried rice, and he stated that his taste had been sitting under a heat lamp. It's kind of stale, okay? Eventually, after a few more cases pop up, an epidemiologist is sent to the site to investigate. As it turns out, that fried rice is contaminated with gram-positive endospore-forming rods. What is the most likely cause of the illness given this information and these choices? You have your hand raised.
Okay, can I answer mine later?
Sure.
And if I get it right, is it B?
It is B. Okay, so we're going to talk about why. So what's your name?
Lexi.
Lexi. Okay, so we got Lexi's taken care of. She answered a question on her own, so that's one thing you guys can do instead of answering your questions later on. So this answer is B. So what we're going to do is break down why it's not the other one. So the information that it's giving us here, really the most important information here in the entire thing, easy, is the endospore, right? And so if you guys are hearing endospore, you know these are bacteria that make endospores. You know it's bacteria because endospores is bacteria, right? And the only groups that can make endospores are Bacillus and Clostridium. Do any of these other choices have Bacillus or Clostridium in their name? This is a way easier question than what you would probably think it was looking at it at face value, because it's got a ton of information in it. But now we can look at this and say, just because they have diarrhea doesn't mean they have salmonella. We have so many, and I mean, I don't think you guys can even begin to appreciate how many things can cause diarrhea, even non-infectious things, right? So we get a question?
Well, I just wanted to say, like, why is the endospore more important than rods? Because rods are just fillers.
So you can be bacillus shaped and your name won't be bacillus. So for example, so bacillus just refers to the shape of the cell, not the name of the organism. And so this organism is actually named bacillus something. We know that that's its name because it's in italics for us. We've got our binomial nomenclature here. So these are the names of the organisms. So when we start talking about shapes of cells, we say that you are a coccus or you're bacillus, or you're, pleomorphic, or you are curved, right? And those are what you could be. Your name isn't going to be always coccus something. Your name isn't always going to be bacillus something, but you will have a shape of some kind. So Escherichia coli is a bacillus shaped cell. It has nothing to do with bacillus in its name, though. So sometimes the names of those organisms can be giveaways. So obviously Bacillus cereus is bacillus shaped, right? That's going to be in their names. And so you can actually also break that down too. If it is named Bacillus cereus, we know a couple things about it. We know A, it's named Bacillus, so we know it makes endospores, and B, it's named Bacillus, so we know it's rod shaped. So you know two things about these guys already. As it turns out, and I've mentioned this previously, but I want to reiterate again. If it makes endospores, it's also gram positive, always. So even if I hadn't said on here that it was gram positive, and I give you all this information, it's contaminated with endospore forming rods. What is it, gram positive or gram negative? Could have been a question too. And you should have been able to tell me that it was gram positive because all endospore forming bacteria are gram positive, right? So that would be an even more like the next level of critical thinking question, right? But here we get a lot of information. If I hadn't just reviewed with you guys the fact that bacteria that make endospores are Bacillus and Clostridium, you might have been racking your brain as to what this could possibly be. And you can see how this would be an easy pitfall for somebody to immediately see salmonella and pick it. Because they're like, this definitely causes diarrhea. But be careful, because the information that I'm giving you, following up with that, be sure that you're collecting all of that information, assessing that question appropriately, and then going through all of your choices to eliminate why they wouldn't fall in. Why isn't it salmonella? It's that salmonella isn't Bacillus or Clostridium. It doesn't make endospores. Why isn't it Escherichia coli? Same thing. Why isn't it Staphylococcus? Same thing. We can also rule out Staphylococcus because Staphylococcus says it's a coccus and not a bacilli. And so that's another way we could do that. So without even having to know that salmonella or E. coli are gram negative, which they are, by the way, you don't even need to know that because we know they don't make endospores, right? And so that's how you can rule out those other guys. All right, so that's the idea with our critical thinking questions. It probably asks an easier question than what the face value would suggest, but it's breaking it down and getting that. So great job, Lexi, for catching that one. Lexi, right? Okay, I want to make sure. All righty, next. We're going to get into chapter 7. We're going to be talking about viruses today. So viruses, they are acellular microorganisms. If they're even organisms, And so this is the debate in a lot of biology as to whether or not viruses are even alive. We'll talk about why that is here in a moment, but these are microbes that are infectious pathogens, whatever they are, that can infect all of the following. They can infect bacteria, algae, fungi, protozoa, plants, animals, anything that's made of cells. We've found viruses that infect it. So these guys are pretty prevalent in the world. There's plenty of viruses that infect your body that may not have symptoms. In fact, a good portion, probably 20 to 30% of your genome consists of old viral DNA that's just chilling doing nothing. That's pretty impressive. And so they don't go away sometimes either. So viruses, what are they? Are they organisms? Are they alive? What role do they play in evolution of life? Like I said, back when our old ancestors were getting infected with viruses, that we still have the DNA in our own genomes from them getting infected, and we've carried it with us. Does it play any role in our active biology? We aren't entirely sure yet. We don't know. And there's plenty of viruses that may be infecting you right now that aren't going to cause you to have a cold or something like that, but could still be invading your cells and having some sort of effect in your biology. In fact, the way that viruses infect bacteria have a massive impact on how the bacteria cause disease. We're going to talk about that too. So there's connections between viruses and cancer as well. If you don't know that, probably most of the women in here will be aware of that. Things like HPV. It's the whole reason for Pap smears, checking to see if you have HPV and if you have any abnormal cells as a result of having an HPV infection. Yes.
I don't think that like the genes, a lot of things like, for example, just using me, Ehlers-Danlos syndrome has roots of a virus in the ancestors.
Yes.
Like people with Romani descent are more prone to Ehlers-Danlos syndrome because of the Holocaust.
Right.
Like,
So, right. So we think that past infections that may have been inherited down the line, that the genetic information from past infections can lead to certain disorders like Ehlers-Danlos or other things like that. There's a lot of evidence supporting that. In fact, exposure to certain things may predispose you to having certain autoimmune diseases as well. So there's a lot going on with viruses and the effects on our body that we don't know about yet. We're still investigating. So there's a lot going on there. If you guys aren't aware about HPV, if you think it's just some sexually transmitted disease that like isn't really super common and that people don't really get, it's actually been estimated that 90 to 100% of sexually active people have been exposed to HPV. Most people clear it naturally. See how quiet it got in here from that. This is not a made up statistic. Most people will clear it naturally from their bodies just fine with their immune system, but not everybody will. And you can have development of these abnormal cells, and your body can potentially still get rid of it, but it's important to keep up with your Pap smears so we can keep track of those abnormal cells and remove them as needed in those smaller settings before it gets to actual true cancer development, and you know, reduce that risk. We still have plenty of people that die of cervical cancer here in the United States, but has been drastically reduced just from screening with Pap smears. So it's really important to consider that aspect of the viral infection side of things as well. Best way to prevent the spread of HPV is to continue to use protection, of course, condoms and barrier methods like that. We'll talk more about the STIs in chapter 24 though. All right. Are these alive or what? So we don't usually talk about viruses being alive. We usually talk about them being active or inactive. And so if we're talking about cleaning agents or things like that that kill viruses, whether or not they're alive is disputable. So we usually just say they inactivate viruses. That's the more accurate way, because these guys are obligate intracellular parasites. This important term is going to come up over and over again in this course. Obligate means they are obligated. They have to be, okay? So they have to be intracellular inside of our cells. And they're parasites because they're feeding off of our resources, using our resources and our organelles and all of the tools that we use in our cell to build our cell. They're stealing them and building their virus copies with it. They don't have any of that machinery to build virus copies of their own, so they steal all of that from their host. So they are obligate intracellular parasites, whether they are alive or not. Well, we've defined a cell as a membrane-bound entity that can make a copy of itself. It has everything that it can do to make a copy of itself. These guys clearly don't have that. They don't have a nucleus. They don't have ribosomes. They don't have amino acids. They don't have any tRNAs to help move the amino acids to the right place. They don't have anything that they need to do that. They have genetics, and they have something to protect those genetics. And that's the bare minimum. And so they just pop their genes in there and let your cells do the rest of the work. And that really is how viruses function. It's pretty impressive. So we're going to talk about those details. These are also able to influence our host genetic makeup in some cases. Even I was lowballing it, apparently. 40 to 80% of the human genome may have remnants of viruses. 40 to 80%. Yeah, I was really lowballing it with that 20 to 30. So put that into perspective, that maybe a lot of your genes is just leftover junk from viruses. And if you think that, well, a good thing that the viruses that we have today don't incorporate their genes into our genome. I think we've already mentioned HIV. Have we not mentioned HIV in this class yet? HIV incorporates its genome into your cells' genomes. Good luck getting it out. We can't. That's the whole point. And so talking about those things and the ability of the viruses to incorporate their genomes into our DNA in some cases, which is also how HPV would function as well. It's going to disrupt your cell cycles and things like that by doing that. But that in their ability to incorporate themselves into your DNA, hiding out there, we can't get it out. We don't have good mechanisms to eliminate that, other than by eliminating just the cells entirely. And so in some cases, that can become a permanent relationship. So you can imagine how that could be passed on from generation to generation. Yes.
So this is kind of, is that why like way back in ancient times, they say, do not disturb my body or I'll give you some crazy.
I don't know that they say that back in ancient days. What does that mean?
Like in the mornings, like do not disturb or I'll come back and unleash like the mummy and stuff.
Oh, like the mummies and things like that. You know, I don't know. Because I think with the mummies and stuff like that, they tried to keep the body as intact as possible because they literally thought that what they had when they moved on was what they would have when they moved on. So they didn't want you disturbing their stuff because they literally thought that like we can keep the body nice and pristine. They'll take that body.
Right? Like we find things.
You could think about it that way. You could think about it that way. That part of it is that maybe part of that thinking is simply that like if people who were healthy might have come in to see dead bodies, that they're likely to get that. And that just gets incorporated into the culture. One good example of that, and this isn't directly related to viruses, but related back to parasites, the worm parasites that we talked about in the last chapter. It's you know highly probable that the reason that maybe perhaps people who are practicing Judaism who don't eat pork as part of their religion, that likely the reason they don't eat pork was because their culture adopted not eating pork due to the risk of parasites being transmitted to the population from eating undercooked pork, which is a high probability if you do eat undercooked pork. And so it's likely that things like that get carried in culture and incorporated into religion or cultural practices. And they nowadays we don't think about that maybe have come from protecting from pathogens. So that's a good idea to bring up. That stuff does carry a little bit. So to put this into perspective, this is a yeast cell, which is tinier typically than what our typical normal body eukaryote cells would be. And this is a little tiny budding off daughter cell. This is what E. coli would look like, about 10 to 20 times smaller than what our normal eukaryotic cells would be. And then we would go down to like streptococcus and what have you, all the way down here to down here where we have polio and yellow fever virus. Okay, so you see these tiny little dots down here? These are viruses, okay, that can infect eukaryotic cells. And tell me polio doesn't have a big effect on your body. Ask FDR what he thinks about that, what he thought about that, right? And the effect that polio can have on your body from this tiny little virus. We had to completely revamp how we were filtering our water as a result of polio because it was transmitted within contaminated water, and how we were treating our water was just not filtering out polio from people passing it through their feces and things like that, and how we were retreating our water and contamination like that. And now our knowledge of how to filter out polio helped us greatly when we had to reconsider how to filter our water from COVID, which was also getting passed through our water and through our feces and things like that. So this is one of the smallest, these two viruses are some of the smallest viruses that we know. Canine parvovirus, another incredibly tiny little virus that we have that can be infectious to people. And you see this little dot down here, just for size comparison. I didn't do a very good job of that, but you guys can see it. That is a hemoglobin molecule. A hemoglobin molecule, not your red cells. That's a hemoglobin molecule. Your red cell is going to be somewhere around the size of that yeast cell, okay? So to put into perspective the size of these viruses. All right, so these guys, they are not cells, they do not resemble cells. They only need a minimum of two things. They need an external coating and they need their genetic information. So literally just two things. The first one, their coat. And the second one, their genome. That's it. That's all they absolutely need. Now they can have other parts as well. We're going to talk about what their extras can be, but this is their bare minimum. All right, so let's talk about their structure. So we have that genetic material that's on the inside. So we know of our, double helix, what it looks like. And we have two sides to our DNA molecule. So that our DNA is double-stranded DNA. So these guys can also have double-stranded DNA. No surprise there. That's what bacteria and archaea have as well, double-stranded DNA. But these guys don't only have just double-stranded DNA. They can also just have single-stranded DNA. They can also have single-stranded RNA. This is like what we would use as our working copy of our genes. So when we have our double-stranded DNA, we'll go and make a copy of it using RNA to make a working copy, our messenger RNA. And our ribosomes read that out in the endoplasmic reticulum to make our proteins. And so these guys can just have that as their genome. And so when they pop their little messenger RNA version of their genome into our cells, we can just start reading it and start cranking out their protein. We don't even need it to be DNA first. So we can start reading it right away. So they can have that. They can also be really weird and have double-stranded RNA, which would also look like a double helix, just like the DNA, but it's just made of RNA instead. So this is really foreign to our cells and how our cells behave, because our cells don't have any double-stranded RNA anywhere in them. In fact, if your body sees double-stranded RNA, it immediately knows this is bad guy territory and can respond without even having to have it specific to that virus. If there's any double-stranded RNA, we know bad guy is there, okay? And this gets even more complicated because when we start reading our genes, your DNA, which is, your ladder, if we had our ladder, separated out like this, one side looks like this and another side looks like this. And then one side we will read to get our messenger RNA that will be read by the ribosome to make protein. And this is the central dogma of everything that we know about genetics, basically. So our ribosome reads this messenger RNA that we made from our DNA, one side of our DNA, to make the protein. And so the side of the DNA that we use to make that messenger RNA is our template strand for that messenger RNA. It's a template, we'll call it template. And that template has the opposite, basically, because A binds with U for RNA, right? And T would make an A, and so on and so forth, for however they're going to be bind G to C and C to G. However, we're going to be writing out our matching code to make our mRNA. We're going to get into details of the genetics of that in the next chapter. But that's the basics of how we would be making our copy of our mRNA, and then using those sequences to decide which amino acids go in what order for our proteins. That's what our ribosomes do for us. So as that's all happening in our genetics, we have our template strand, which I'll put a T under it just so we can remember that that's template. I'll put temp. And then this one, code. Because if our mRNA is the A becomes the U, and the T becomes the A, and the G becomes the C for our mRNA, the other side of the DNA actually says pretty much the same exact thing as our mRNA. So that's our coding strand. So it actually says the same code as our messenger RNA. So we're starting to see the point of having the two sides of the DNA, and we're starting to see what happens when we make our mRNA. Well, this mRNA is what we would call positive sense, because our ribosome can just straight up read it. It has the code that the ribosome needs to read. So this gets more complicated because our single-stranded RNA for our viruses can be positive. We could just read it with our ribosomes, or it could be negative. We might have to make a copy of it in order for our ribosome to read it. And so this is,
read the negative one.
you read the positive part. Yes. So your ribosome would literally be able to latch onto a positive RNA and spit out protein. That's the idea. Negative RNA, you would have to make a inverse copy of it. Your A to U and your U to A, and G to C and C to G in order to be able to read it with your ribosome that way. Okay. Don't ask me why they have positive or negative. Don't ask me why some of them are double-stranded or why some of them are DNA or RNA. It's just how they've evolved to be. Okay. And as it turns out, your single-stranded DNA ones can also be positive or negative, okay? So we've got literally, I just told you they can have 6 different genomes. Yes.
A virus has double-stranded RNA,
Uh-huh.
and we can't read double-stranded RNA. Does the virus not infect us?
No. So it has double-stranded RNA. We can't directly read the double-stranded RNA, but we can separate it and then read the strand. Yes. So if we see it in our body anywhere, the double-stranded RNA, we can see it just automatically and know that it is foreign. But if it gets into our cells will still separate it and still read it. Okay. And so you will start to see that I just told you guys we have 6 different genomes. So what were our 6? We had our DNA ones and we had our RNA ones. We have double-stranded for each of these possible, a single-stranded positive sense, a single-stranded negative for each of these. So they can have 6 different kinds. All of our genetics for all eukaryotes, all of our genetics for our prokaryotes, our bacteria and archaea, always double-stranded DNA. But viruses, they have all these different versions of that genome. And part of the reason that they have that is so that they can't be targeted by our immune system. Part of it is to make it quicker and easier when they get into our cells to start making their products. And part of it is just how their other aspects of their machinery are going to relate to their structure and things like that. Because while the minimum of one of these viruses might be just that genome coded by some protective protein, some of these guys have a lot more going on inside of them than just that. Okay. So this is our bare minimum, having our genome types, having your genome. And for example, if you are HIV, you would have a single strand positive sense RNA genome. Okay. I just told you HIV incorporates its DNA into our DNA. And then I told you that its genome is RNA. So you can see how this is going to get complicated. So we'll get to that. But anyway, so these are the possible genomes that we can have. Come on, guy. I'm trying to draw here. All right. Nope, I want to erase this. Nope, I can't just do it that way. Anyways, so that's our basics, is having a coat around that. I'm trying to find my eraser here, so bear with me. I wish I should just erase all this really fast. And that coat is always going to be made of protein. And that protein coat is called a capsid. That's what I'm trying to write up here. And it is always protein. All right. And what do we know about DNA and RNA? What does the NA stand for in these guys? Nucleic acid. Right. Good job. So we have a protein coating around a nucleic acid genome of some kind. That's the base for a lot of these guys. If you are only capsid plus your nucleic acid, the word for this is your nucleocapsid. And if you're only that, then we call you naked. Easy enough? All right, because some of these guys, and you can see it on the left in this picture, right? Some of these guys have an envelope around them. So here, this fancy triangle business, you can see the genome in the middle in the pink. That fancy gray triangle business, that's our capsid, and it looks fancy like that, okay? It sorts itself in these icosahedral sort of orientations on there. That's the nucleocapsid. And everybody has a nucleocapsid. But some of these guys also have a double membrane around them. So if you have a membrane as a virus, that's called an envelope. Okay? And then you're not naked, exactly. We call that enveloped. Or to remind you guys, that's gonna be like a version of clothed, right? So we're clothed with a membrane. So we're clothed with this membrane. And where do you guys think they get that membrane from? The cell, exactly. So the cell that they are inside of, so they're making copies of their genome, they're coating themselves with their protein, and then your nice membrane is on the outside. you know, keeping them inside of the cell, protecting them from the outside world. So what shelters and keeps our stuff inside and keeps us outside, they're just gonna push through it. And as they're pushing through that membrane, they cover themselves, just like if I had a big old giant blanket and you just walked through it and it would swaddle your whole body in that nice cozy blanket. They are swaddling their whole nucleocapsid with the membrane of the host cell. And that's how they're going to envelope themselves with the host membrane. Okay? So those are enveloped viruses. Some of our guys also may have some other proteins or enzymes they carry with them. HIV is one of those. It has an important one we're going to talk about in a little bit. That helps it go from single-stranded RNA to DNA. Okay. All right, so capsid, that's our protein coding our genome. Envelope, if we have one, if we are an envelope virus and not a naked one. So we talk about polio. Polio is a naked one. It doesn't have an envelope. Okay. Most of the really, really tiny ones don't have an envelope. Parvovirus doesn't, for example. So these capsid guys may just be capsid and the genome inside. But if we have our nucleocapsid that is also coated by a membrane, then we are enveloped. That's the membrane they stole from the host. And then we have spikes as well. So if you're a naked virus, your spike, as we are going to show in our next slide, I'm actually going to go to the next slide. So if you're naked, your spikes are going to be part of your capsid. They're sticking out of the capsid. Okay. If you're enveloped, your spikes need to be on the outside of the envelope. Why? Because the spikes are what they're going to use to get into your cell. They are the key to the doors on your cells. Okay, so spikes are the key to the doors on your cells. And some of these guys will use doors on your cells that are very specific to certain cells. And some of these guys will use doors into cells that we can find those doors on a lot of different kinds of cells. For example, rabies virus. Rabies virus is able to infect a very large number of mammalian species and cell types of those species. Okay, so it can get into a lot of different animals. Whereas if we look at herpes, human herpes virus, like herpes type C, herpes C can only get into human livers. Right, so that's it. It's not going to infect your dog's skin cells. Right, so some of these guys can be very specific, and some of these guys can be more general. But it all depends on your spikes, your keys, and what doors you're opening with those keys. So depending on what proteins they have as their spikes, these are usually glycoproteins. So what does glyco tell you? Sugars are involved, right? So as part of that structure, remember with protein, shapes are a lot more important than the actual chemical makeup. So the structure, the shape of that key is really important for getting in and out of cells. So these are normal things on the outside of cells that allow cells to do their jobs. That they will normally recognize a molecule and say, hey, come on in, you're here to help with this thing that I do, or that thing that I do, or you help me recognize that you're another cell, or whatever it is. And these guys mimic that, and that lets them get into the host cell that way. Does that make sense to you guys? You're starting to get that? All right, so your capsid, your outer protein protective shell, is made up of these subunits called capsomeres. So a capsomere are the many subunits that are going to make up the capsid. All right, you can have two different shapes, generally speaking, of capsid, helical or icosahedral. And that kind of speaks for themselves, right? So let's look at some examples. Our helical capsids, the capsomeres just circle around. So you'll have your genetic information, which might be this pen here. And they just assemble the capsomeres helically around it. Just like it shows in the picture, right? So that's our helical capsids, all right? Sometimes that is enough. And that's all you would need. You could be like a rod of just genome covered by helically by capsid stuff, right? So that's an example of that would be tobacco mosaic virus, which is a really common one for tobacco. It's really well studied genetically as far as the genetics of viruses and what we know about viruses. But we don't see a lot of eukaryotic viruses that look like that. But you could also take that nucleocapsid, and you see this guy right here. Let's see if I can zoom in. This guy here, you see how inside of that, we've created a nucleocapsid that has a helical shape to it. And then we've taken that helical guy and let him be a little worm inside of the membrane. So he's all curled up inside of the actual membrane around it. So we have a helical capsid wrapping up our genome and just wound it up and shoved it inside of an envelope. All right, so sometimes they'll be inside of a more round envelope. Sometimes that envelope can be kind of around the whole rod shape. And it just sort of depends on the virus. These guys have crazy different shapes, crazy different sizes. Ebola, for example, helical capsid with a membrane, but it looks more rod-like. It has a shape that we call a shepherd's crook. It looks sort of like this. Oh, that's not what I was trying to do. Okay, well, whatever. It looks like this. But it's all enveloped and it's got that helical capsid on the inside and everything. So these guys can get pretty crazy as far as how they're going to utilize a helical capsid. Whereas our icosahedral capsids, these guys, we can see electron micrograph here, so it's using an electron microscope, shooting electrons at things to see them at the atomic level, that these would actually be triangles that are just basically stacked up on each other in this sort of a format, okay? And they do that. And so these capsomeres, these little ball guys, will come together to form these sort of subunits of the capsid and then build the whole capsid out of that. Okay, so we have the two shapes. We have the helical and the icosahedral. We good? You can put envelopes around any of that. So bacteria, these guys are ridiculous. Tell me that they're not. So bacteria can be infected by viruses as well. We already said that. If you're a cell, you can be infected by a virus. And they get the fanciest viruses. Look at this. This is not a cartoon down here. I realize that the one next to it is a cartoon, but that with the star on it, that's an actual colorized electron micrograph of one of these viruses. So this is a bacteriophage. Bacteriophage. You'll often hear me call them just phage. A phage or bacteriophage is a virus that infects bacteria. And what these guys do, it is nuts. See that how its genome is contained in its icosahedral head? All right, so, and you see this, they call it the sheath, but it looks like a spring sort of wound up down there. And we've got these, what they call tail pins. And then they've got these things that they call tail fibers and the collar. Man, tell me that doesn't look like legs. Like it's a big old spider with a big old head on the top of it. But what these things do is the legs actually sense that, yes, I have landed on the right bacteria. I've landed on E. coli. So that their legs serve as their key and recognize that the door that they're opening is to E. coli, right? So the legs come down and land on E. coli. They say, this looks right to me. It causes the protein to change its shape. So it squats down and puts its little booty down on there. So its little booty, I'm talking about its tail pins here. That clearly is going to shove those spikes down into the E. coli. And then that spring sheath is going to contract as a result of all these proteins changing their shape, contract down and inject that genome straight into the cell. Is that not just so cool? And so that's how it gets the genome in. These guys don't have to enter the bacteria. They just inject their genome into the bacteria. And that's how all phages are going to work. In bacteriophages, they all always inject their genome in there. They might not all look exactly like this, but they all sort of look crazy. And so these guys are going to work that way. Yes, question? So after they inject that genome, the bacterial cell is going to look at that and say, oh, this is DNA. I know what to do with that. Because even remember, do bacteria have nucleus? No. So already all the machinery that they need to make copies of the genome and to make the products of those genomes and everything, it's all right there in the cytoplasm. And they're ready to go. So they'll just start making copies of that and making the products of it and start building up these new viruses. So just automatically, immediately just start making new bacteriophages. And they'll just keep doing it until the cell explodes. And they'll just explode. So I've already told you, our viruses will step out of our cell and like nicely, comfortably blanket themselves in our membrane and then walk off into the ether all happily. And then you can see how your cells could recover from that. But bacteria cannot recover from this. They're going to bust open. Okay?
I don't think I could recover from that.
Yeah, no. Lucky our cells don't usually behave that way. So usually our viral infections don't involve busting open. So this is just showing us a picture of some viruses that might have an envelope or don't. This is just kind of displaying that. So moving on to that viral envelope, that membrane that we steal from our host cells. Usually, most of the time, if you're infected with something that is enveloped, so let's say you're infected with COVID, okay? And you are making COVID viruses. As those viruses are budding out of your cell membrane, they've replaced all of your surface proteins that are normally on there. The ones that would help out with recognizing cell-to-cell contact or recognizing molecules that are supposed to go in and out or all that stuff, they'll replace all of your surface proteins with the viral spikes. So that way, when they push out, they have their viral spikes already on their surface. And they just cover themselves with a pretty blanket with their pretty design all over it that they already put on there. So they cover your cells with their spikes, which can cause a problem because your proteins are on the surface of your cell for a reason, right? They're doing jobs for your cell. So this can cause all sorts of problems with your functionality of your cells. We're replacing all of those regular membrane proteins with the viral spikes, right? So spikes are essential for attaching your virus to the host cell. But now you can kind of also see that if you're covering my infected cell with viral spikes, that the cell next to me that might not yet be infected is going to be attacked by those viruses. The proteins don't know what they're doing. But those that normally are keys to the cells, they see the cell next door as this protein interacts with the door next door. And so now you have fusing of infected cell with the neighboring cell because of those spikes being on the surface. You have the inability to attach to the surfaces that we're normally attached to because our normal proteins that attach us aren't there anymore. And all this. So we have detachment of cells. We have formation of these giant multinucleated messes because they're all fusing together because of the spikes on the outside. Those are called synthetia. So you've heard probably that term before. You guys know what RSV stands for. Respiratory syncytial virus, right? Synthetia. They form those giant multinucleated cells because the spikes cause them to come together like that. All right, moving on, because we've got a lot to get through still. All right, so that's the idea. We've already talked about the specificity of these guys, but they can be very, very small as well. Some of them only have the genes that they need to make copies of their genome and then the proteins that they're made out of. And that's pretty much it. But some of them carry extra genes. So the size of their genome can vary. We know that it can be DNA or RNA. It's not going to have both. It'll be either DNA or RNA, and it's always going to be one kind. So, you know, COVID might only have its type of genome, which I don't know what it is off the top of my head. Apologies. We know that HIV has positive sense RNA. We know that influenza has DNA, or they're never going to switch in between. Okay, so that sort of stuff. We talked about positive and negative sense. So we already mentioned about how our DNA is DNA, double-stranded DNA. This is really good drawing of it, but you guys get the idea. And it's double-stranded. And that normally our genetics go from double-stranded DNA to what we call mRNA, which is single-stranded. And that this is our working copy of our gene. And then to protein, which is read by our ribosomes. Right? And we'll talk about the details of how exactly that happens later on, but this is the central dogma of genetics, that this is the rule. You go from DNA to RNA to protein, but I have literally already told you that HIV has an RNA genome, and it's going to make DNA and put it in yours. So it's going to be reverse direction of what everything we always knew about biology would say. So it's going to go this way. This process normally is called transcription. So HIV contains an enzyme called reverse transcriptase. ACE means enzyme, typically, okay? So this enzyme makes DNA out of RNA. This was a revolutionary discovery. We had never seen anything like this prior to HIV and investigating it. And now we can use this as a tool in molecular, genetics and things like that. And we'll learn about that in chapter 8, but we can use that as a tool to allow us to take RNA and then go back into DNA for our own study purposes, things like that for science, right? So these guys can have special enzymes like that. Yes.
Is there anything else that has that?
Yes, there are other viruses that have that. So we have a whole grouping of viruses called the retroviruses. Retroviruses will have reverse transcriptase, and they can also borrow our tRNAs. We'll talk about what they do later on in chapter 6, but they all can do that. And so there's other viruses like that. HIV is our most obvious one, but actually hepatitis does it. Hepatitis B does it too. And hepatitis B, interestingly enough, isn't a DNA virus. So it's a DNA virus, I know. This is what I'm saying, like, this stuff gets really complicated. It's a DNA virus that will go DNA to RNA to protein, just like we do. But from its mRNA middle guy, it makes DNA from that to make its copies, and it also puts that DNA into your genome to lead to liver cancer and things like that. Super complicated, and I'm aware of this. So there are plenty of viruses that do that, and you guys don't need to know all of which ones do what. We'll talk about some of the ones that we need to know in detail when we get to those viruses. Right now, we're focusing on those concepts. For animal viruses, these are the main steps for animal viruses, okay, our eukaryotic viruses. We have these main steps, these 6 steps. Adsorption, so this is attachment. Penetration, we get into the cell. Then we uncoat. We have to expose our genome so we can actually work with it. Remove that nucleocapsid. Then we have synthesis, so making more capsids and genomes, making copies. And then assembly, put together new viruses. And release. This can be from, we can bud through the membrane. This is for our enveloped guys, right? They have to do that. Or you can be exocytosed. That's in the name. Don't let it be a crazy word for you. Cyto means cell, exo means outside. So literally it's taking a bunch of these viruses and just putting them outside the cell. And generally our cell doesn't want them in there anyway, so it's happy to exocytose them. But it's what we do whenever we're making things that need to be excreted from our cell anyway. So this is a normal cell process, exocytosis, putting things outside of the cell, moving things outside. So these are going to be our naked guys. Okay, so with animal viruses, these are the steps they would follow. We clear on that? So we go through each of these steps. Again, adsorption, not absorption, adsorption. Attaching to that cell, that spike protein is going to match its door and be able to get in. If it doesn't match, it can't get in. Okay, so this is going to determine our host range. So our host range, the people or the animals or the creatures that we can infect, is determined by our spike's ability to get into whatever the doors are on that cell. Does your key fit the lock? And so, like we said, hepatitis B infects only the liver cells of humans and no other things. Whereas when we look at broad range viruses like rabies, infects various cells of all mammals, which we mentioned previously, right? Moderately restrictive, polio infects intestinal and nerve cells of all primates. So a little bit more vague than what we have with Hep B versus with rabies. While we're on the topic of rabies, okay, while we're on the topic of rabies, I would like to send out, an APB or whatever, however you want to look at this. Rabies is easily the deadliest virus that we've ever discovered in man, easily. If you guys don't know that about rabies, you're fooling yourselves. So the reason that we vaccinate our pets is, of course, to protect our pets, but it's also to protect humanity. And rabies is pretty rampant, a lot less so nowadays in wild animal populations. In particular, here in Oklahoma, we're talking about skunks and foxes and coyotes and things like that, maybe raccoons and other things. But once you get rabies, you have a very short window of time to help your immune system out to get rid of it. And once you start, if you get bitten by an animal, it takes a while to get symptoms, maybe a month. But if you get bitten by an animal and that saliva got into your wound at all, and you start having any symptoms whatsoever from rabies, you are dead. There's no helping you. 0% chance of survival. It is 100% lethal. There's nothing. There are not survivors of rabies. I want to be very clear of that. So if your kid is playing outside and they're like, oh, a raccoon was out there, ha ha ha, it bit me, and they have a little bitty scratch on them, to the ER right away, give them their shots. We can give you antibodies that will help fight right away, as well as we'll give you them immunizations to build up their own immunity to keep fighting it off so that it doesn't get embedded in your nervous system. Because once it gets into that nervous system, you are donezo. And it's the same with animals. If anybody here work at vet clinics? Yeah, so you've probably seen it. If an animal comes in and they suspect rabies, we're putting it down. We don't even investigate. We're not going to test it. So we'll give it some time to make sure that it's actually exhibiting the signs of rabies if we think it's a pet. If it's not a pet, we're probably just going to put it down. And so we don't play with rabies. So vaccinate your pets. keep your community safe. If anybody you know gets bitten by an animal that you even remotely suspect is at all rabid, go to the ER, get your shots, okay? Because once you have symptoms of rabies, it is 100% lethal. It is one of the only viruses that we know of that is 100% lethal. You want to know another one that's 100% lethal? You guys already know, we already talked about it. HIV. If you don't get treatment for HIV, you will die from it. So some of these viruses, we can actually bank on 100% lethality if they're untreated. And we know we can keep HIV under control, but as soon as you stop your medications of keeping from replicating that virus, it's just going to start replicating again, and it will take over your immune system and eventually kill you. So how these viruses infect and how they infect the body are all important to consider whenever we're talking about all these diseases. Yes, you're fine.
But you died from AIDS, not HIV.
So you don't get AIDS without HIV.
Right.
Right.
But.
And
AIDS is
HIV
mature HIV.
Right. You don't ever have HIV and not get AIDS if you're untreated. Always HIV progresses to AIDS if you are not treated. Always. So if we get HIV, it'll always progress to AIDS if we don't get treatment. And you always will die from AIDS as a result of that. And so AIDS, yes, it's a syndrome that develops from having an HIV infection, but HIV always progresses into AIDS if we aren't treating it. So good distinction. So, but you won't have AIDS without the HIV. So
But you can have HIV and not have AIDS.
no, not if you're not. Yes, if you're getting treated, but only if you're treated. So I want to be clear about that. So this is really important distinction, right? You can have HIV and have treatment for it and just keep it as HIV positive. Once you're HIV positive, you're HIV positive for life, period. That's it. Even if you're undetectable levels, sorry, you're still HIV positive, okay? So yes, so you can have HIV then and not develop AIDS, but only if you're treated. If we stop treatment, we will immediately revert back to beginning that process of developing AIDS. So that can't be stopped without these medications. So I want to be very clear about that that's only controlled by the medications that we have available to us. And what do those medications control? What do you think one of the things is? Reverse transcriptase. Yeah, reverse transcriptase. Easy target, because we don't have that in our cells. But it's not good enough once it gets incorporated in your cells, right? So that's really important. So good delineation between the two, but it's really important we can't unmarry them. All right, so we already talked about uncoding a little bit. You dock with your cell, and you can either just merge your membrane and release your contents in, or the cell will eat you up, and then you'll release your contents. So that's what this is talking about here. If you're eaten up, that's endocytosis. This can happen because of phagocytosis, or just because you docked with the right lock, and now the cell's like, yay, come inside. Or you will just literally just merge with your membrane, and everything that you have inside will just spill inside. So those are two ways that can happen. Synthesis, this is just making copies of your genome. So if you are an RNA virus, you can just release your little happy RNA in here, and we can start reading it right away. We can if you're positive, we can read it right away and start making proteins like our capsomeres and our spikes. We can also make a negative sense and use it as a template to make more positive sense. We got to get it from somewhere. It doesn't just make copies positive to positive. You have to use something as a template. And so then we would build our parts and build new viruses that would then exit the cell appropriately through either budding, which is what this is showing here. This is budding, or exocytosis, we'll talk about later. If you're a DNA virus, it's a little bit more complicated because everything that we have in eukaryotic cells for DNA happens in the nucleus. So now you have to get your DNA, if you're a virus, you have to get that DNA into the nucleus, right? So we're going to take that viral DNA and move it into the nucleus through a nuclear pore. And inside of the nucleus, we'll be able to make copies through replication of that DNA. We'll also be able to go from DNA to RNA to protein like we normally would with that viral DNA, okay? And then we would be able to build everything either within the nucleus, or we could do some steps of it outside of the nucleus. It just depends on the virus. You can even bud out of that nuclear membrane. So this is an image of a nucleus. This is the nucleus of a cell. This is the cellular membrane of that cell. Normally your nucleus isn't this large. All these little dots in here are new virus particles. And these weird crystal looking structures, it's just masses and masses and masses of proteins to build the new viruses. So that whole nucleus has been just relegated to making virus. And this is pretty much what happens in your cells if you're infected with the virus. Your whole cell will be designated to making virus. And you won't be able to do anything else. So now you can start to see why you start to feel like crud when you get sick, right?
When they make medications, they have to make the medications and do the kind of thing.
If-
For
Yes, for antivirals, right? So it has to be able to get into the cell and get to the right compartment of the cell to have its action, correct? For antivirals. And it's also why now you can start to see how these viruses could be so different. You can start to see why antivirals are so specific to viruses. I mean, we don't really have many antivirals out there that are blanket antivirals, do we? It's mostly like Tamiflu. What's it for? The flu, right? So a lot of antivirals are like that. The HIV antivirals, they're for HIV. They don't really help much with any other viral infection because these viruses work so specifically for what they need to do. We can target that virus. Also, the hard part with these guys, with bacteria, we can target how they make their cell wall. We don't have peptidoglycan cell walls. So stop them from making peptidoglycan and you're good. That's what all the cillins do. Penicillin, amoxicillin, and all of those guys. And that's easy targets, right? But viruses, they use our equipment. So you can't stop them from a lot of things. So you have to find something unique about them to target with our medications. And that gets hard because they use our cells and our equipment in order to do all of that, right? So is this starting to make you guys feel like viruses are a little bit scary? They probably should. All right. So moving on, when we release these guys, we have our budding. We've already showed our budding. But you could also go through exocytosis. I don't have a good picture of that, but it's literally like at the Golgi apparatus where we move everything in the vesicles around the cell or out of the cell or secrete it. It would just be getting a whole bunch of viruses in there and just pushing them out of the cell. The vesicle will do the work for us, okay? So you don't need a membrane for that to happen. But what I'm telling you is most of the time with animal viruses, they don't bust out of the cell like the bacterial ones do, okay? All right, obviously with all of this stuff going on, taking over our machinery, making all of these structures inside the cell that's just like designated for the virus, putting all of these keys on the outside of our cells, our spikes as keys, trying to get into the doors next door and merging with the cells next door, or covering ourselves with keys and no longer the normal proteins on our cell surface that do normal cell surface stuff. Now we are no longer attaching to things. We're no longer behaving the way that we should. And that leads to cytopathic effects. And that's really what cytopathic effects are. Things that I could look at your cells under a microscope and say, oh man, we're screwed. Okay. And so things like this can actually lead to a development of certain effects of disease. With Ebola, for example, the fact that it covers your cell surfaces with so many of its proteins that your cells are going to detach from the surface. They're going to merge with each other and behave weirdly to such an extreme extent that the fragile, tiny little blood vessels of your capillaries and things like that, they become fragile, even more fragile, because the cells aren't holding them together anymore. So your blood leaks out. And it starts small. But then that damage becomes worse and worse and worse. And suddenly you're bleeding out of your eyes and your nose and every orifice of your body. Right? And so you can see how this would happen if we're affecting so many cells on your body with these viruses taking over basically how your normal cells work. All right. Some of these guys, like we talked about with HIV, can incorporate their genomes into your genomes. And when they do that, that HIV genome that is now incorporated into your cells is called a provirus. It's not a virus fully, right? It's just its genetic information, but it's still viral genetic information. We call that a provirus. Okay, this would lead to a chronic latent state if we're not actively making viruses from that and it's just hiding out. We see this, for example, with like herpes. All right, so it's hiding out in your cells. It's not making active expression of infection, but when your immune system gets stressed out because you're stressed out, we start having replication of that virus and making more virus. And so that is when we can actually see that cycle going back and forth between hiding out and making more virus. So that latent state, whenever you still have that viral DNA in you, it's just not making virus. That's a chronic latent state. All right, and we've talked about how they can incorporate in your genome and disrupt your cell cycle, like what we see with HPV. We estimate that about 13% of cancers, and it's probably more than that, but we have evidence to support that about 13% of human cancers are probably caused by viral infection or related to viruses of some kind. Now, it leads to unchecked growth of the cell. With our bacteriophages, these guys can also have double-stranded DNA or other versions. And again, these guys will inject their genome into the bacteria. They only infect one species of bacteria. So this T-even phage, like T2 and T4, they only infect E. coli. Bacteriophages only infect one bacterial species and not any others. So you have to have a different bacteriophage to infect Salmonella than what we do with E. coli, right? So these guys inject their genome in there and then eventually lead to lysis. This is an electron micrograph of a bacterial cell that's being basically attacked by multiple bacteriophages and even growing bacteriophages inside of them. It'll break open and release a whole bunch of these guys. They cycle between two stages, sort of like our viruses do. So we've talked about how, HIV can hide out in your cell, or it can make virus, and so can herpes hide out in your cells, or it can make virus. These guys, for the bacteria, do the same thing. When we're making virus, we call this our lytic cycle. When we're hiding out, that's our lysogenic state. And so when we have what we would have called provirus, we here call prophage, because it's the phage, hiding out the genome of the phage inside of the bacterial genome. And it'll just wait like that. These guys can actually carry extra genetic information with them, things that can code for toxins or antibacterial resistance and all sorts of stuff like that whenever they hide out. And they can switch between these two states. Anytime you can switch between two states, we call you temperate. So temperate phages can switch between the prophage or the lytic cycle, making more bacteriophages, okay? So you can switch between the two. When we switch back to the lytic phase, making more virus, that is called induction. When we carry extra traits and we have become lysogenic. So I remember that lysogenic is when the genes are incorporated in because the gen. So lysogenic genes from the phage incorporated into bacteria. They can have extra traits conferred to them. When that happens, that's called lysogenic conversion. The whole reason that diphtheria causes disease, the whole reason that cholera causes disease, the whole reason that botulism causes botulinum toxin is because they have these traits conferred to them by viruses that they got infected with. That's called lysogenic conversion. So we have viruses to blame for a lot of the bacterial diseases that we have. And I mean a lot of them. All right, if we're trying to grow our viruses, we can't just put them on agar, right? Because they can't grow by themselves. We have to give them cells to grow in. And so that would lead to the development of tissue culture. Obviously, we know how viruses can cause disease in humans. Some of them have high mortality rates. Some of them give a common cold. Most of your common cold effects are going to be caused by various cold viruses. We know about 200 different cold viruses plus. And so these are usually caused by viruses, but these can also lead to long-term disability, like effects that we see with like polio. And then we can actually use something called interferon, which is going to signal your neighboring cells that you're infected to help your body, deal with certain viral infections. So that's a broad use drug that we can use for that sort of stuff. Then we have our non-cellular infectious agents, just really quick. We're going to run through these guys. These are prions. Prion are proteinaceous infectious particles. These are just misfolded proteins. So we could take a protein that normally might fold like this, and then we have all of the inside of this guy, all the inside of him might be hydrophobic. And then become misfolded. So now we have a whole lot of the hydrophobic on the outside. Well, obviously, in our normal body, hydrophobic does not want to be on the outside because we've got a lot of water in our cells. So these guys will stick together. And so hydrophobic sticks to hydrophobic. And so this misfolded protein, you might just get one in your body. It comes to your normal protein in your body, causes it to misfold like this. And they all just aggregate together and make each other to build off of each other. You come into contact with one of these misfolded proteins, and it comes into contact with your normal proteins. You could have this buildup going on for like decades. You eat a piece of beef in England in the early 90s, and you are hanging out 30 years later, you come down with brain issues. It could be bovine spongiform encephalopathy or mad cow disease. So we call this variant form of mad cow disease variant Creutzfeldt-Jakob disease, okay? And this leads to spongiform encephalopathies. So spongiform because it's causing sponge-like effect in your brain. And it's an encephalopathy. Encephalo means brain, pathy means we're having disease in the brain. Okay, so sponge-like disease in the brain from those aggregated misfolded proteins. And it leads to eventual death. These are also 100% lethal. And it's just a misfolded protein. There's no genetics, there's no cell, there's no membrane. It's just a misfolded protein. Obviously not super common, but these proteins are pretty resistant and can be spread pretty easily. So Creutzfeldt-Jakob disease is the biggest one. There are other ones we'll talk about in later chapters. And we don't really know the exact way that this happens or why or how, but we do know it's misfolded protein that causes more misfolded proteins. There are genetic information that you might have in your cells that'll make you more susceptible to these misfoldings.